This is part 3 of 3 for bacterial structure videos. So in this video, we're going to finish the last few structures that are associated with bacteria. I want to make note again that these structures are not necessarily given to every bacterial species, but rather are some of the perks that bacteria can possibly have. And we're going to see that as we compare and contrast the following two structures. The two structures that I want to compare and contrast are capsules and endospores. So the reason I want to compare and contrast these two structures are because these structures are going to have the same generalized function. And the generalized function of these two structures are going to be for protection. However, the protection that they are going to be used for are going to differ based upon the environment. So capsules and endospores, as I mentioned, for protection, but capsules are going to be used for bacteria that are found inside of the body. Therefore, the protection that they're offering is to protect the bacterium from the immune system. The immune system is going to include white blood cells, which are going to have phagocytosis in order to try to destroy bacteria. So this protects the bacteria from phagocytosis. Endospores, on the other hand, are found outside of the body. And they're going to be used to protect against harsh environments. So the three harsh environments they need to know are heat, chemicals, and dehydration. The next thing that we need to understand with capsules and endospores is the state of the bacterium. So in a capsule, it's found inside the body, and so the way that I think about that is the bacterium wants to be in the body, and so this is going to be a happy state, or in other words, the bacterium is going to be active, or in fancy scientific terms, vegetative. Because it is active, it needs nutrients. And so a capsule is going to allow nutrients to get through. Or in other words, it's going to be what we call permeable. Although things can get through, they have to be very small. So capsules are very helpful in also causing disease because many antibiotics or many chemicals have a hard time getting through the capsule. It's the small nutrients they're able to get through. And so capsules are also helpful in causing disease because they protect from phagocytosis and they also don't allow chemicals to get through them. But they do allow some things to get through because they need to get those nutrients. Endospores, on the other hand, I think, okay, a bacterium isn't happy when it's outside the body, so this is going to be in what we call a dormant state. And because it is in a dormant state, it doesn't want things to get through and ruin its genome while it's sitting there. So an endospore is protected against heat, chemicals, and dehydration so that the genome isn't damaged, or the genomic material. And so in order to protect its genetic material, it's not going to be permeable. The next thing we need to understand is what they are made of. And so capsules are made of a sugary substance which is called Glycocalyx. And Glycocalyx is going to allow it to have a very smooth layer. So if you're in the lab and you grow bacteria, you notice that colonies, when they grow on a plate, have different appearances. And if you aren't, and you look in the textbook, you can see different appearances of bacteria that grow on different types of media. And so this is going to actually give it like a smooth look. And so the Glycocalyx is going to be very smooth, and it's going to make it very slippery. And that's why it allows phagocytosis to not occur. Endospores, on the other hand, are made of peptidoglycan. But this is many, many layers of peptidoglycan. So although gram-positive, if you remember, with the bacterial cell wall has a thick peptidoglycan or many layers, the endospore is going to have approximately hundreds of layers versus the approximate 30-ish layers that you could find in a gram-positive. So there's a lot of peptidoglycan to help protect it. Because of that, we need to understand what type of bacteria make capsules and which make endospores. Looking at endospores, because we just talked about the amount of peptidoglycan necessary, the only type of bacterial species that can create that much peptidoglycan are going to be gram-positive. So these are going to be only gram-positive. However, we need to make sure that remember that not all of these things are associated with every single type of bacteria, and so it's not going to be all gram-positive, only a select few. On the other hand, because Glycocalyx is just a sugary substance, both gram-positive and gram-negative are able to create the sugar and secrete it. So there's some species that have it that are gram-negative and others that are gram-positive. So these are the comparing and contrasting between capsules and endospores. So although they both have the generalized function of protection, if you dig down further, you're going to see that they have a lot more to it. The last thing that we need to understand is the creation and the leaving of an endospore. So I'm going to draw a line to help separate the capsule, and this bottom area is going to be how a bacterium would enter and how it would leave its endospore. So if the bacterium is outside, it's going to be in an endospore. However, as soon as it enters the body, so for example, let's say tetanus, which is an endospore forming bacterium, if you step on a rusty nail and it has an endospore on it, it will go into your body. Once that endospore gets in, the tetanus doesn't want to be in its endospore or its dormant state. It wants to be in an active state. So it's going to break out of that endospore and become a vegetative bacterium. And that is what we call germination. So when it breaks out, it's called germination and becomes in its vegetative or active state. Now let's say with time just by chance that the bacteria falls back out of your wound. And so as it goes back out into the regular environment, it doesn't like that. And so it's going to form its endospore again. And this formation of the endospore is called sporeulation. So again, the breaking out is called germination, while the formation is called sporeulation. So this is all the information that you need to understand with capsules and endospores. If you need to write down this information, or you still need to draw some of the pictures, I recommend that you pause the video now. The next structures that we're going to be talking about, and last structure, is going to be a biofilm. So biofilms are something that you find when you have a group of bacteria. And so with biofilms, you're going to get a bacterium that's by itself, but you're going to get many of them that come together. And that makes that bacteria that has its friends around it a lot happier. Imagine if I'm going against an army, I'm going to lose if I'm single. However, if there are many of me, I'm going to have my own army, and so therefore I'm going to have a higher chance of surviving. That's exactly what's happening with the biofilm. The bacterium alone will lose against the other army, which is the immune system, but if it has its own army, it's going to be able to survive. In addition to having this group of bacteria, they're going to secrete a sugary substance to help them adhere as well. And so the biofilm is going to give two things to the bacterium. The first thing is that it allows colonization, or in other words, they're going to be able to stay alive and continue to grow, and adherence, or in other words, the ability to attach, and the other thing is that it allows cell-to-cell communication. So it seems very strange, but bacterial cells are actually able to talk with each other. So because they're so close together, they're able to talk through chemical gradients that are being secreted. Imagine if I am in the same room with you and we are talking face to face. It's a lot easier to understand, we can communicate, we can react to the emotions, and we can really understand each other. Now let's say that you decide to move out of that room and you either leave the house or just leave into another room where you can't really hear me. As soon as I start talking, you're not going to be able to get that understanding. And so you can't really react to what I'm saying. Even if I'm saying things that are necessary, like, hey, you need to move over to the right a little bit more or move to the left, you can't understand that. But with bacteria, they're going to be able to adjust to their environment because they have these other friends, these other bacteria around them to communicate with. But when they're alone, they're not going to be able to really communicate and therefore cannot adjust as easily, which leads to them dying more easily. So this is the purpose of the biofilm. Know that it allows colonization, adherence, and cell-to-cell communication. So with the structures wrapping up, the adherence part is very interesting and good to know, is the fact that we have fimbriae that we talked about in the first video, we have pili, which we talked about in the second video, and now we have biofilms, which we talked about in this video. So there are three things that bacteria can use for adherence, or in order to attach to a surface. And so I just make note of that. And these are also commonly known as adhesins. And we'll talk about that term more in the future when we talk about bacterial pathogenesis. But I just want to make mention of these things. And the last structure that we're going to talk about is going to be the ribosomes. And all you really need to know about it is that it's for protein production. So if I was to draw a ribosome, it would look something similar to this. And the ribosome has a total weight of 70 S. The 70 S, S just refers to the way they measured it. They spin it down really fast, or in other words, it's called centrifuge, and the centrifugation is going to allow it, based upon its distance, they measure it. So strangely, the two parts, there's the large subunit and the small subunit, are not actually going to measure up to 70 S. The large one weighs 50 S, while the bottom weighs 30 S. And all you need to know now is that it's meant for protein production. And to make note that these ribosomes are different than eukaryotic ribosomes. And as I mentioned in other videos, these ribosomes are going to be necessary to target in one of our methods of control when we are using antibiotics. So these are all the bacterial structures they need to know. And if you missed the other two parts, I recommend that you go to part 1 and part 2 in order to get the rest of the material.